Okay, class, we are now moving into chapter number 6 of our textbook, and this chapter focuses upon the cell. And hopefully, you have been introduced to the cell in previous science classes, perhaps in classes in middle school. The cell, a lot of times, is not the most fun chapter for us to discuss. However, it is a very important chapter as we are dealing with biology. Of course, we are studying the science or the study of life. And because of that, we do have to take some time to deal with the concept of the cell. Before we move on, however, into this chapter, I do want to run across to you a concept known as the cell theory. And there are three components to the cell theory, which is important for you to understand and important for you to grasp as we move on. Since we are studying biology or the study of life, we do need to be aware of these three principles or these three aspects of the cell theory. The first of those is that all living things are made of cells. So if we're dealing with something that is currently living or something that was living in the past, we are dealing with something that is made of cells. Non-living things are not made of cells. The second component of the cell theory is that all cells come from existing cells. And what we mean by that is that cells do not just arise out of the air. In order for there to be additional cells, there must have been a cell that began that process. So first, all living things come from cells. Second, or all living things are made of cells, rather. And then all cells come from existing cells. And then the third component is that cells are the basic unit of structure and function of an organism. And what we mean by that is the cell is actually the lowest level that we can go to in which all of life's processes are carried out. Now, if you remember from chapter 1, we began the process of the hierarchy of organization. And we started at the base level of the atom, and then the atom built into molecules. Those molecules then are going to build into what we call our organelles, and organelles build then into cells. The organelles in and of themselves are not the smallest level that is living. Now, they carry out specific functions just like the organs inside of our bodies do. But in and of themselves, we would not consider them living and able to carry out life's essential processes. But the cell is able to do that. In fact, if our cells are not respiring, if they are not reproducing, if they are not digesting, then we on an organismal level are going to have complications and difficulty in that. So keep those three ideas in mind as we progress and begin moving through this chapter detailing the cell. The first thing that we need to come to terms with from the idea of the cell theory, if we look at that third component, and that is that cells are the basic level of structure and function for all organisms, we have to understand that there are two basic types of cells. We have what are called prokaryotic cells and what are called eukaryotic cells. And this is where we now get into the concept and the idea of our different domains. And the domains that we talked about back in chapter 1, there are three. We have bacteria, we have archaea, and then we have the eukarya. And when we're dealing with prokaryotic cells, we are dealing with the bacteria and the archaea domains. And then when we are dealing with plants, animals, fungus, or protists, then we are dealing with our eukaryotic cells. Now, there are 4 to 5 basic features to all cells. In the AP biology book, we see that there are these 4, and then there's one additional from a text that I have also been utilizing in classes. So the first is this plasma membrane. Another name for that is also the cell membrane. The semi-fluid substance called the cytosol. Another name that a lot of times we will use for that is the cytoplasm. There are also chromosomes. The chromosomes are going to carry the genetic material, also the DNA. And then ribosomes, and ribosomes are important for the production and the manufacturing of proteins. And then the fifth, if there would be a fifth that we would put down here at the bottom, it would be a cytoskeleton. And the cytoskeleton is important because it helps the cell, whether it is a prokaryotic cell or a eukaryotic cell, be able to hold its shape. It provides some basic structure to the cell in much the same way that our skeleton inside of our bodies provides stability and provides structure and the ability to move of the human body. So again, if we were going to look at those 5 things, we would say that all cells, whether prokaryotic or eukaryotic, are going to consist of a cell membrane. They're going to consist of cytoplasm or cytosol, DNA, ribosomes, and also a cytoskeleton. So let's begin with the prokaryotic cells. Now, when we talk about the prokaryotic cells, we're dealing with types of cells that are very different from the eukaryotic cells, very different than animal cells, very different from plant cells. These are 4 characteristics that they have in their cells. Number one, they have no nucleus. And what we mean by that is they don't have a membrane that surrounds their nucleus. In fact, we call this region a nucleoid. Now, a nucleoid would be similar to the nucleus in a eukaryotic cell, with the exception of that it is unbound. There is no membrane surrounding it. Therefore, we do not call it a nucleus. There are no membrane-bound organelles. So as we progress through this chapter and we talk about things that hopefully you remember from some of your past classes, mitochondria, endoplasmic reticula, dealing with Golgi apparatus, those types of things, we're not going to find those in prokaryotic cells. And then they have a cytoplasm that is bound by the plasma membrane of the cell membrane, which is very similar to what you will find inside of the eukaryotes as well. And I'm going to move through these slides very quickly. There's a lot of graphical slides that will be in your PowerPoint presentation that I will be sending to you. And so if you have any questions on these, please let me know, and we will talk about them in class. But here we see a typical rod-shaped bacterium, and you can see the basic components of this. And then we have two different pictures of this. One, we have a schematic, and then number 2, we have this microscope view. But you can see here some of the components that we would find that are similar to eukaryotes. There's ribosomes in here, which are important for protein production. Here's the nucleoid, in which we find the chromosomes or the DNA, very different from the type of nucleus that we as eukaryotes possess. But ribosomes, the nucleoid for the DNA. There is a plasma membrane that you can see here, surrounded by a cell wall. Now, animal cells, human cells, we do not have cell walls. Plants do, but we do not. So a cell wall is not going to be something that all cells actually consist of. And then there are some what we would call organelles that are found inside of them. And here we have what are called the flagella. And flagella is going to be necessary for movement. But like I said, I'm going to move through these fairly quickly. So prokaryotes, not a whole lot to discuss because they are very simplified, just with those basic regions that we have just discussed. Eukaryotic cells, however, are very different. They typically are much larger than your prokaryotic cells are, and they're going to do a lot more things than your typical prokaryotic cell is able to do. So first of all, we have a nucleus that is bounded by a membrane. This membrane is very similar to the cell membrane that goes around the cell, but only instead of dividing cell from cell or cell from extracellular material, we're dealing with a membrane that is going to separate the inside of the nucleus from the cytosol or from the cytoplasm. Membrane-bound organelles, this is where we get into discussions of things like Golgi apparatus, mitochondria, and those various types of organelles. And then the cytoplasm is the region that is located between the nuclear membrane and the cell membrane, and it's in this area of the cytoplasm that we actually will find the organelles. Now, the cell membrane or plasma membrane, it doesn't really matter which of those two terms that you use. What is important for us here is really one term, and that term is called selective permeability or selectively permeable. And what this means is that this barrier is going to determine what things are able to move from one side of the cell membrane to another side of the cell membrane. And so it is going to serve as the border patrol, if you will, of what is able to pass from one side to another. Some things will move readily. They will freely move from one side to another. Some things are going to be controlled. Perhaps they've already got enough of that material in the cell, or enough of that material has already left the cell. But those are things that we will get into as we move along. And the cell membrane is actually composed of a double layer of phospholipids, and we typically will refer to this as a phospholipid bilayer. And what we have occurring here, if you remember from our discussions in chapter number 5, when we talked about phospholipids, is phospholipids are a different class of lipids. Most lipids are going to be hydrophobic. They want nothing to do with water. However, your phospholipids have a hydrophilic and a hydrophobic portion. And so what happens here is, since you've got 70 to 95% of your cells interior and 70 to 95% of your cells exterior that is going to be composed of water, the phosphate head, or the hydrophilic head of a phospholipid, will actually point towards the water, and the tail will point away from the water. And then you will have a second phospholipid that will point, say, into the cell, so that where the water is where the phosphate head is going to be pointing, and then the tails will point towards the outside. And so what you end up with is this structure that looks kind of like this. If we were to say that the back of my hand, where the metacarpals are located, if we were to say that this was the phosphate head, and this was the phosphate head, and my fingers were the tails, then what you've got is you've got one that is pointing to the exterior part of the cell, which is where a lot of water is, and the tails are pointing in. You've also got, on the other one, you've got the phosphate group that's going to be pointing into the cell, and you've got the tails are pointing out, and so you get this structure that looks kind of like this, okay, where the tails are pointing inside, and then you pointing toward one another, and then the heads are pointing opposites as they're pointing towards the water regions. And your slides, you will see this. Here is a graphic representation of that, in which you see the phosphate heads pointing out of the cell and into the cell, and the tails pointing towards one another. Metabolic requirements will set the upper limits on the size of cells. One of the things that we need to bear in mind is that cells are going to grow in such a fashion that their surface area to volume ratio is the most efficient. And what we mean by surface area to volume ratio is we're going to see how much area is going to be, let's say, along the top and along the bottom, as well as the thickness, as compared to the entire three-dimensional structure. And what we want is we want to have a great surface area to volume ratio. Okay? We want to have that because the more volume it has, then we're going to see that there may be some nutrients that are unable to get in, or waste that are unable to leave because the cell happens to be too big. So we want to have that good surface area to volume ratio. And surface area will increase by a factor of n squared, whereas the volume will increase by a factor of n cubed. So the larger the cell is, the greater the increase in the volume. The volume is always going to be able to increase at a greater rate than the surface area is. So we want that to be as low as possible so that the cells are able to do what the cells are supposed to do. And we'll do a few examples of this when we are in class. So here we look at eukaryotic cells. They have membranes that partition the cell into organelles, and in plant and animal cells have a lot of the same organelles. So here we look at a typical eukaryotic cell, and you can see there is a cytoskeleton here that is able to give structure and stability to the cell. Here is the nucleus, in which you find some chromatin, which is the DNA material. You get the nucleolus, which is going to be utilized to build ribosomes that will then move out into the exterior part of the cell, or the interior part of the cell rather, so that the proteins can be manufactured. And you see a bunch of these other things: Golgi apparatus, lysosomes, the peroxisomes, mitochondria, rough ER, smooth ER, and we'll get to these as we go along. But this is what we would consider to be a typical animal cell. And then here is what we would consider to be a typical plant cell. Now you can see that this cell is very different from an animal cell. However, there are a lot of organelles that are very similar. You can see that there is a nucleus, there is nucleolus, and there's a chromatin. There's rough and smooth endoplasmic reticulum, a cytoskeleton, ribosomes, Golgi apparatus, so a lot of the same structures, but there are some key differences, one of those being this central vacuole, which is going to be a storage facility. Animal cells, human cells do not have these. Chloroplasts that are going to be utilized for photosynthesis. Animal cells do not have chloroplasts. And we also see these cell walls, and cell walls are also going to be things that we as animals do not have. And if you remember a couple of chapters ago, well actually chapter 5, when we were dealing with some of the polysaccharides, this is where one of those polysaccharides really comes into play. Cellulose is going to give some great structure and stability to these cell walls, which is something that we as human beings do not have inside of ours. Okay, so let's now begin moving through the cell. I'm going to move through this rather quickly, so jot down questions that you might have as we look at each of these components of the cell in kind of a fast-forward fashion. So number one, let's start with the cell's nucleus. The cell's nucleus is going to be important really for a couple of major reasons. Number one, the nucleus contains the DNA. Okay, now there is some DNA that will be found in some other places. For example, in animal cells, there is going to be mitochondria that contains its own DNA, and inside of plant cells, the same thing will happen in the mitochondria as well as in the chloroplast. But for the most part, we're going to find most of the DNA to be found inside of the nucleus, and found in a particular region known as the chromatin, which as we move along in our chapters, our further chapters, we will learn that this chromatin will condense into chromosomes before cell division takes place. And ribosomes are also a key component because they are going to be manufactured or produced inside of what is called the nucleolus, a really dense portion of the nucleus that contains RNA and proteins, and this RNA and proteins come together to form the ribosomes that is necessary for protein production. The nucleus is the brain of your cell. It is what tells the cell what to do. It controls all of the activity of the cell in much the same way as your brain controls all of the functions of your body. So your body wouldn't behave the way it does without the brain. Your cell wouldn't behave the way the cell does without its nucleus. There is a nuclear envelope. The nuclear envelope is very similar to the cell membrane. Its job one is to contain the nucleus, just as the cell membrane contains the cell. And things are not able to move from the nucleus into the cytoplasm unless it goes through nuclear pores. And there is a bilayer that is there, a double membrane very similar to the cell membrane as well. So you've got the lipid bilayer that is very similar to the same type of thing on the exterior of the cell. Nuclear pores are what are going to allow the ribosomes as well as the messenger RNA, when the messenger RNA has been produced inside of the nucleus, to move out so that protein synthesis can actually begin. Pores are going to regulate the entry and exit of molecules, just as there will be channels that will determine what moves in and outside of the cell. So if you can understand the cell membrane, the nuclear membrane is not going to be a difficult concept for us to be able to understand. Again, you've got the chromosomes that are inside of the DNA that actually form from chromatin. So when chromatin actually condenses, when it gets thicker, becomes thicker stranded, chromatin then becomes the chromosomes, and that is where the DNA of the individual is actually located. And then we have the nucleolus. The nucleolus, again, is found within the nucleus, and this is the site of ribosomal RNA synthesis, rRNA synthesis, so that ribosomes can be produced. Now, what exactly are ribosomes? Well, ribosomes are particles that actually allow proteins to be produced. Without ribosomes, we would not be able to produce the proteins that we have. And here I'm dealing with both types of cells, prokaryotic as well as eukaryotic, because ribosomes exist in both of those types of cells. Without them, we wouldn't be able to synthesize proteins. Now, they are located in two different regions. We have what are called freely floating ribosomes, which are located just in the cytosol. Almost, you could kind of picture this as if the cytosol were a big swimming pool. Then imagine beach balls floating inside of that swimming pool. Well, the beach balls could be reminiscent of ribosomes. So ribosomes are freely floating inside of the cytosol. It'll be like beach balls floating inside of a swimming pool. Then there are others that are bound. These are actually connected to what we call endoplasmic reticulum. When they are connected to endoplasmic reticulum, we call this type of endoplasmic reticulum rough endoplasmic reticulum. So you have two different types. There's rough as well as smooth. Rough contains ribosomes, smooth does not. Okay? And so here you can see a diagram of a ribosome. It's got two different subunits, the large subunit and the small subunit. And these are going to be very, very important when it comes to the manufacturing of our proteins. Okay? So now again, we're going to just kind of breeze through this as much as we can, talking about these various organelles that we have. The endomembrane, endo meaning inside, so we're going to be inside of the system, regulates the protein traffic and also performs the functions of metabolism inside of the cell. So what is the endomembrane system? What is this system that is located inside of the cell? Well, it consists of these following organelles: the nuclear envelope, endoplasmic reticulum, both rough and smooth, Golgi apparatus, lysosomes, vacuoles, especially if we're dealing with the plant cell, the plasma membrane, which is the cell membrane, which is the exterior part of the cell that dictates what comes in and what is able to leave. And we also have what are called vesicles, and we'll talk a little bit about vesicles. Their prime importance is transporting things from inside the cell to outside the cell, or vice versa. They're going to kind of act as the taxi service. They're fluid-filled, kind of like fluid-filled bubbles or substance-containing bubbles that move things inside of the cell. So let's look at the endoplasmic reticulum first. This accounts for more than half of the total membrane inside of the eukaryotic cell. So there's a lot of endoplasmic reticulum in there. They are continuous with the nuclear envelope, and what we mean by that is they just they keep moving from the nuclear envelope. There's no separation. It is a continuous membrane in which we are able to find things that are going it's a transport system is essentially what it is. Two distinct regions. There's smooth and rough ER. Smooth, no ribosomes. Rough is going to be studded with ribosomes. So here we have a picture of that. You have some rough ER. You can see all these little dots. This rough ER, ribosomes makes rough ER. And then here at the outline portion, you can see some endoplasmic reticulum that doesn't have any ribosomes. This would be the smooth ER. Okay, so what is the difference between them? What do they do? Okay, smooth ER will synthesize lipids. This means that it helps to build lipids. The fact that it doesn't have ribosomes means that it cannot do anything as far as proteins are concerned. It metabolizes carbohydrates, so it's going to go through those chemical processes. When you think of metabolism, just think of chemical processes. Okay? Metabolism is typically defined as the sum total of all chemical reactions going on inside of the body. So that's what we mean by metabolism, going through chemical reactions. Detoxifies drugs and poisons, and it also stores calcium ions. And calcium ions are going to be very important, especially when we're dealing with muscle cells, because it's calcium that actually will allow us to contract our muscles. Without calcium, we wouldn't be able to do that. Rough ER, bound ribosomes. These are going to secrete glycoproteins, and glyco meaning sugar, protein being protein. So these are going to be proteins that are actually covered or bonded with carbohydrates. Distributes transport vesicles, which means that the proteins are actually going to be surrounded by these vesicles so that these proteins can move from one area of the cell to another area of the cell. And we call it the membrane factory for the cell. So this is where we're actually going to continue to build the various components of our membrane. Golgi apparatus, this is a series of flattened membranous sacs. The functions modifies products of the ER. So the examples that I typically give with this, the application I give of this, is that we have a warehouse. The ER has produced what we want. Then what will take place in the Golgi apparatus is, whether it's a lipid, whether it's a protein, whatever the case may be, whether it's coming from smooth or it's coming from the rough ER. When it arrives at the Golgi apparatus's function is to take what has been delivered to it and modify it. Once it has been modified, once we know exactly what we want and it is exactly in the form that we want it to be in, it is going to be packaged, and then once it's packaged, it is then transported. It is placed inside of the vesicles. So if we wanted to use this as an example, a warehouse receives our orders when we call them in, we telephone them in, we send it in online, whatever the case may be. Then that order goes to a manufacturing facility. In this case, it would be the endoplasmic reticulum. Endoplasmic reticulum designs and it builds to specification what we have ordered. That is then going to be taken to the final portion of the warehouse, where the order that we have called in is modified so that what goes in the box is exactly what we ordered before it is placed inside of a truck, whether it be UPS, FedEx, or the United States Postal Service. Use whatever example you want, and then brought to the destination. In this case, it would be our home. Same thing here. The Golgi apparatus receives the material, it modifies it, it packages it, and then it's going to ship it out in the form of a transport vesicle so that it's able to move through the cell to the site specified by the nucleus. We have lysosomes also inside of our cells. Lysosomes are fluid, digestive enzyme containing vesicles. And their job is to digest macromolecules and also to take care of any unwanted material from the outside. So if something comes into the cell, as it comes in, it may be something that the cell does not necessarily need, necessarily want. Perhaps it's a harmful substance. then a lysosome will fuse with that vesicle, and those digestive enzymes will break it apart. When we think of the word lyse, we think of something breaking down, and that's what lysosomes do. They are able to break those things down. They hydrolyze, which means that they're through the process of hydrolysis. Remember that from a chapter ago. When hydrolysis occurs, water is used so that things will break apart. Proteins, fats, polysaccharides, and nucleic acids will be broken down thanks to our lysosomes. Some types of cells will engulf other cells in what we call phagocytosis, and what this is it's just the ability of this cell to actually wrap itself around and engulf a particle that is there so that it is able to destroy it. So lysosomes will fuse and destroy whatever is there and whatever is not necessary. And here is a picture of something like that actually going on. Something has come in through the process of phagocytosis. A lysosome will then fuse with it, and it begins a process of destroying everything that was actually contained within that particular vesicle. Vacuoles, something that plant cells and fungal cells have that animal cells do not. These vacuoles are actually going to be storage facilities. They are derived from the endoplasmic reticulum and the Golgi apparatus. Food vacuoles are formed by phagocytosis when these things are brought in. There are also what are called contractile vacuoles, and then central vacuoles. And the central vacuoles will hold organic compounds and water. And these are going to be found in a lot of our mature plants. And here you can see a picture of that central vacuole that is there, a major storage facility inside of our plant cells. And now as we look at this as a review, then we're going to talk about some other things in the next lecture. So this first lecture is just going to deal with this endomembrane system as well as the key components to every cell. As a review, the endomembrane system is a complex and dynamic player in the cell's compartmental organization. And keep in mind that the things here that we are looking at are going to be the endoplasmic reticulum. We're going to be talking about the nuclear membrane because that is continuous with the ER. We've got the smooth ER. We've got the Golgi apparatus with our vesicles. And then you have the cis, which is going to be the closer to the ER. The trans is going to be on the outside. You can almost look at it this way. The cis is the receiver. The trans is the transmitter going to transmit out the Golgi apparatus, the modified package material that is now ready for delivery. Here we have the plasma membrane. So what would we consider to be this endomembrane system? It is going to run from the nuclear membrane to the cell membrane and contain all this network between those two things to try and get a particular macromolecule, a particular substance formed, and then move it out of the cell so it can be utilized by the body. We're going to pick up in the next lecture dealing with concept 6.5, which talks about mitochondria inside of animal cells, mitochondria and chloroplasts inside of our plant cells. And so if you have any questions concerning the endomembrane system, any questions concerning the cell theory, or any of the organelles we've covered thus far, please write those down, and we will cover those in class in the coming week.